I'm going to talk about aging Earth, but actually it's the aging populations of Earth that I'm going to talk about. And I'm going to look in particular at the effects of increasing longevity in the countries of the Earth. I'm in a sense continuing the recent shift from the past and the origin of life into the future and what it holds. And I want to look particularly at how one can avert what will be, if we can't do it, it's an imminent plague and it's a dreadful condition that we face with neurodegenerative diseases. So let me introduce the topic. Since I've used the word plague, I will go back a little bit to mention some of the plagues of the past. Most of you probably know that in the Bible, there's this description of the plagues of Egypt. They may be mythical, otherwise they're just over 3,000 years ago. There were these sort of different plagues. If you want to interpret what they could be, it's maybe an early example of climate change. These are thought potentially to be red algae, a bloom of those caused by changes in climate, and then climatic changes in the climate and so forth, of winds and so forth. And so you can begin to think there may be a reason for these plagues if they existed. A bit more recently, most of you will know about the Black Death. It's one of the bubonic plague that went through Europe in the 14th century. It killed perhaps half the population of Europe. Here's a couple of sufferers. Here's somebody trying a probably not very effective remedy of tossing some things into the air. It had a good side. Boccaccio wrote the Decameron in Florence. But again, we know why this took place. It's again change of the human populations of the Earth. The numbers of people went up. The trade routes were increasingly used. And this caused the spread of disease throughout the world. Perhaps one of the least well-known plagues is Spanish flu, maybe a version of bird flu. It took place in 1918 to 1919. It's estimated to have killed 50 million people. That's more than the entire fatalities during the First War. It affected particularly young men who probably had extremely poor health, having been in the trenches for some years. And so these were the plagues of the past, but modern medicine, antibiotics and antivirals, have transformed the situation. So, for example, penicillin, I think, saved 100,000 people in the Normandy landings. There are, of course, fears about new outbreaks of infectious disease, Ebola and so forth. You read about it all the time. But by and large, modern medicine, public health, vaccination, so forth, are really effective at controlling these classic plagues and treating other forms of common illness. And this, of course, causes an increase in lifespan. And I thought I'd just put this little picture up. One of the consequences of it, one of my favorite cartoonists, as we contain infections, improve hygiene, nutrition and so forth, our life expectancies increase, and so other illnesses start to become prevalent. Heart disease, cancer, and most recently Alzheimer's, that, at least in my generation, brings the same fears of those of the contagious diseases of the past. So this is a cartoon. It's a retirement age protest. The retirement age is being increased, and the call is, what do we want? Why do we come here? And what were we saying? Which reflects some of the well-known symptoms of Alzheimer's disease. Now, one of the very interesting things about Alzheimer's disease historically is that it was a 20th century curiosity. It was first described in 1906 by, of course, Alzheimer, and he studied a 51-year-old woman who was in the Frankfurt Asylum, and this was described as a very unusual case of cognitive failure and memory failure and so forth, of dementia. 51 is not a huge age nowadays, and in fact we now know that she was suffering from tissue which was kept from a familial form of Alzheimer's disease, which is an early onset. But until then, it really didn't figure as it was a curious disease. And it's quite interesting to look at the disease statistics from just say the last 50 years. These are from the US. It's just the disease rate per 100,000 people. It's a log scale. So this is 1, 10, 100, 1,000, going from the late 1950s up to about 10 years or so ago. And the interesting thing is, first of all, there are no infectious diseases here. They've been abolished, at least from wealthy countries. Top of the league is heart disease. But what's interesting is the incidence has decreased by a factor of 5 over 50 years through improved lifestyles and the treatments which are now available. By contrast, if you look down here, in 1980, Alzheimer's disease wasn't even registered. People just got old and forgetful and so forth. It was just thought to be a part of aging, which it is not. And then from 1980 onwards, the number of reported cases has gone up by a factor of 100. Some of this was diagnosis, but still the numbers are increasing dramatically. And so this is really the complete outlier in human disease. Another way of looking at this is everything you can look at in terms of money. And this, again, says why this is becoming a 21st century plague. This, again, data from the US. This is the cost in billions of dollars of Alzheimer's disease in the US, the estimated cost. So in 2015, it was thought to cost the US economy $200 billion. That's in lost working hours, care, and so forth. But the dramatic thing is, the prediction is that by 2050, when most of you will still be around and paying lots of tax, it's going to go up to $1 trillion. If you do some sums, it means the US will spend $20 trillion in the next 30 years or so just looking after people with Alzheimer's. This is not treatment, this is looking after people. If you add up all the world will spend something like $100 trillion in the next just over a generation if we don't do something about this this condition. It's a lot of money. I looked up the annual exports of China, for example, are $4 trillion. And so why is this increasing? Well, it's increasing because longevity increases. In particular, at the age of 65, you have about 1 or 2 in 100 chance of getting symptoms of Alzheimer's disease. At the age of 80, 85, you have a chance of 1 in 3 or even 1 in 2. That's the same risk as getting the Black Death in the 14th century. You're a bit older normally when you got it. Of course, this doesn't apply to scientists who are completely immune from this. So doing a PhD in science is absolutely what you need to do to avert this. So another way of looking at this is that if you then look at the number of people over the age of 65, which will increase dramatically in the next 20 or 30 years, it's been estimated that of all the people in the history of the world who've reached the age of 65, well over half are alive now. And so this is a completely different demographic situation from anything that the world has faced before. This is aging Earth. Many of us think of Alzheimer's disease as something that just affects wealthy countries, but in fact, it's an imminent and global challenge. It's predicted in 2050 that the number of cases will rise from 40 million to 135 million, and 70% will be in low or middle income nations. And you can see, apart from sub-Saharan Africa and parts of West Africa, it's going to affect pretty much the whole world. I wouldn't dare show this in Denmark. For some reason, Denmark seems to have the wrong color here, but I think there must be a mistake. I'm sure that Denmark is just as healthy as everywhere else. So what is Alzheimer's disease? Well, it's a so-called protein misfolding disorder. And I thought I'd try and explain what misfolding disorders are for proteins. Although many of you will know this, I just thought I'd say why protein molecules are so important and what their characteristics are. If you look at a cell schematically, it's pretty close packed with molecules. It's something like 30% solution. If you don't think that's impressive, go and try and make up a 30% solution in the lab. We've got about 100,000 different sorts of protein, if you include those products of gene splicing and so forth. Everything that goes on in the cell is determined by proteins, catalyzed by or controlled by or or the structures involve proteins. And so with other biological molecules, they're packed together in the cell. This is a bacterium. It could easily be any other type of cell. And what's remarkable is that through evolution, their surfaces have evolved, or the surfaces evolved through evolution, so that they will interact with the molecules they need to interact, but more remarkably, they don't interact with the vast number of other molecules in the cell. They remain effectively independent because of this beautiful design through evolution of their surfaces. So what are protein molecules? I just thought I'd remind some of you what these are. They're polypeptide chains, typically 50 to 500 amino acids. There are 20 different varieties, as you know. They're joined in a specific sequence that's unique to every protein. It's encoded in genomic DNA. If you're not impressed by those numbers, you could produce 10 to the 300 proteins from those sequences, different sequences, and I think there are 10 to the 80 particles in the universe, so it's a big number. And biology has selected a certain number for particular purposes. They're synthesized on ribosomes, very large molecular machines. We now know the atomic structures. The mRNA comes in, and from that, the code is translated into the polypeptide sequence, which emerges through a tunnel. But I've got this visual aid. I sometimes say I borrowed it from my wife. My wife would not be seen dead in this row of beads. But it comes out of the ribosome as a string of beads. You can see that I think there are only 8. There's an order of these different amino acids which have different shapes and chemical properties. In order to function, they have to fold up to bring these groups together in a particular way. And so they fold up into very compact structures. And if you look at some of these, this protein folding, how they do that is encoded in the sequence, another marvelous result of evolutionary biology. The left one is myoglobin, the first protein whose structure was determined. This comes into helices and brings together the key residues. Other proteins have beta strands, which end up in beta sheets. And this is acyl phosphatase, an enzyme that typically has this mixture of sheets and strands. If you looked at it in a space-filling model, these would be closely packed. So these are very simple proteins. When I started looking at protein folding, this is what we were dealing with. Things just got worse as time went on because we know proteins are highly complex. And this is actually the protein generating energy for the cell. It was mentioned earlier. If you look at this, you think evolution's pretty powerful. It can take 500 residues, and every time, essentially, it will fold it up into the correct structure. But of course, it's a very select sequence. There are 10 to 300 to select from. This is one that has the characteristics of folding into this state. So folding and self-assembly are the essence of life. Every type of protein, every complex has to assemble correctly. So what happens if proteins misfold? Well, because they're so important, all sorts of things happen. And there are actually many diseases from cancer to diabetes, which are now known, increasing numbers every year associated with misfolding. What happens? Well, this is schematic. If they misfold, some can be degraded because they're not passing quality control mechanisms. Cystic fibrosis is a classic example of that. Some end up in the wrong place in the cell or the organism, and there are diseases associated with that. But the most interesting ones, I think, are ones where misfolding ends up with the surfaces being incorrect when they misfold, and so they clump together and aggregate. And there's a whole set of misfolding disorders associated with aggregation. So what I'm going to talk about from now on, if I use misfolding, the consequences are going to be aggregation. Molecules of the same type will come together to form structures, higher order structures. And some of these are called amyloid deposits. Historically, they were discovered by using the same dyes as we use for starch, and starch is amylin, so they're amyloid. They're, of course, proteins, not carbohydrates. And there are about 50 diseases associated with amyloid deposition. They include Alzheimer's disease, the transmissible prion diseases, but they include type 2 diabetes, where the aggregates form in the pancreas, while these form in the brain, and there are others where there are different even medical conditions can induce amyloidosis by changing the balance of proteins. Each is associated with a particular protein or fragment of protein that's a dominant contribution to these clumps of aggregates of form. And as I said, they include many. Parkinson's, Huntington's disease, motor neurone disease are all misfolding disorders. They have interesting variations in characteristic. Alzheimer's disease, there are deposits as amyloid plaques in the brain. These are very famous. There are also tau tangles intracellularly. These are extracellularly. And you know the symptoms of these. They kill neurons, and the brain shrinks, and we get all the symptoms which are well known in this disease. But they're very different diseases. For example, it's now recognized that many cases of heart failure in elderly people are caused by the fact that amyloid, these aggregates that clump together, change the properties of the muscles in the heart to induce failure. There are even cases, really mutational cases, where the perfectly ordinary proteins, this is actually lysozyme, a protein we were looking at from its folding characteristic, and you can get kilograms of proteins depositing, this is a young boy, in the liver, the spleen, and kidneys, and so forth, and it's fatal. So what's interesting from a structural point of view is that when you extract these deposits from different diseases, which have all these different symptoms and locations, and different proteins involved, they have a remarkable characteristic that they're all fibril-like or largely fibril-like. They can be microns in length. They're a few nanometers in diameter. They've got lots of beta sheet structure. And when we realized this, and others realized this, we were immediately intrigued to know what this other form of structures, this universal sort of fibrillar structures that appear in these diseases. So we had a look at the nature of the amyloid fibrils. And like everything we did, things happened by accident. We were looking at the folding of an ordinary protein, nothing to do with disease, in the test tube, actually an NMR tube. We took it to low pH. We were going to then take it back to physiological pH and watch it fold. But while we were doing this experiment, it turned into a gel. We actually knew that gels were associated with aggregation. And so we looked into the NMR tube, and it was full of these fibrils that we previously thought were only associated with this family of diseases. So we realized that proteins, perfectly innocent ordinary proteins, could convert into these structures. We did a lot of work on this. We looked at many. This is myoglobin, perhaps the least likely protein to form amyloid fibrils, you might think. It's helical, while fibrils are sheet. But if you put it under mildly denaturing conditions and leave it for a bit to look at what's in the bottom of the test tube as opposed to what everybody else does, take what's in the top, you've got these fibrils just like in disease. Everybody said, well, you're just not smart enough to recognize the sequence that codes for the structure. And so a very persistent graduate student looked at homopolymers where all of these beads are the same. And he found that if you got the conditions right, you could generate amyloid fibrils that look just the same again as those from disease. So unlike our functional proteins, where their structure is encoded in the sequence, these are not encoded in the sequence. They're a characteristic of the polymer property of polypeptide chain. So it's a generic polymer structure, and these are the ones we know and love from biochemistry. But there's another structure. This is a model. You can see it actually involves the chain not folding up into compact structure, but actually going into strands, which then come together to form a beta sheet, and they end up forming these long fibrillar structure, double sheets, in fact, which can twist around each other. And this is called a cross-beta structure because the strands are across the fibril axis, which goes on, as I said, for microns. We know the structure of these now. Here's one example we did. These are the strands, and these yellow ones are hydrogen bonds. So actually they're very, very stable because they have this array of hydrogen bonds which holds together the main chain of the protein. And because the main chain is common to all proteins, they look pretty much the same for every sequence, that vary in detail. So what does this mean in terms of the origins of these protein misfolding diseases? Well, another thing that was discovered is that These are not necessarily the most stable thermodynamic forms of proteins. Often these very strongly hydrogen-bonded forms, the fibrils, the amyloid structures, are actually thermodynamically more stable than these. But fortunately for us, and I'm commenting on this in a moment, the rates of conversion are usually extremely slow, so we don't turn solid very quickly. It takes quite a long time. Why are they bad? Well, you lose function. You don't really want kilograms of this stuff in your liver. But it turns out as well that there are, of course, steps in forming the fibrils. You have one molecule, it clumps together with another, and another, and another. If you look at the beginnings of this, you can see these so-called oligomeric structures under the electron microscope, which are sort of little spherical globs before they eventually convert into fibrils. And it turns out these precursors, these intermediates, can very rapidly penetrate cells, even if these proteins are nothing to do with disease. It's a generic property, again, of the polypeptide chain. And so aggregation doesn't just abolish function, disrupt organs, but it can generate extremely toxic species inside. So unlike a bacterium coming from the outside being toxic, these are protein molecules which clump together to form toxins within the organism. We think there's a really unified view of this. Normally, actually, this tendency to aggregate is picked up by so-called housekeeping quality control mechanisms, chaperones, other species. They prevent the accumulation of protein aggregates by targeting them for degradation or wrapping them up and keeping them out of trouble. And it's when these fail or are overcome that we get these large accumulations of aggregates, or we get toxicity and cell death. So if you look at the recent prevalence of these misfolding disorders, they're all modern diseases. They weren't known really more than 100 years ago. And they're all associated with such things as some genetic traits, but largely aging, Alzheimer's, there are a whole range, Parkinson's, motor neuron disease and so forth, which are all age-related, Alzheimer's being the dramatic example. There are also dietary changes. When you become obese, you change the balance of hormones and proteins, and these can aggregate and cause cell death. And actually it's estimated now that there were essentially no cases 100 years ago. Now it can be estimated that nearly 10% of the world's population is suffering from one of these diseases. And at the moment, they're all effectively incurable and largely untreatable. Let me just show you a little movie that I like. It's a way of testing these ideas, because we took the protein associated with Alzheimer's disease, put it in fruit flies, and then we made some mutations that would make it more likely to clump together or less likely to clump together. And very small changes. We knew how to do this from our in vitro model systems. And what you see in a minute is 6 test tubes. The middle one is the sort of wild-type protein. Here we made a mutation to make it less aggregation-prone, and here we made mutations to make them more aggregation-prone. I like this picture a lot because you see you bang the flies to the bottom, you get graduate students to look at them climbing up, and you get this wonderful line. You get the line without even having to plot it. The points do fall off it if you go there. But what's wonderful is this is biology. 20,000 genes in fruit flies. This is physics, changing the properties of these proteins by 1 or 2%, and it goes from being complete health to death. It's really quite remarkable. So in the last part of my talk, I want to say, can we use the information we've known? I'm giving just a sort of flavor of the types of things that have been going on in this field. Can we develop rational therapeutics based on such ideas? Well, how were therapeutics developed in the past? Well, this is a picture from medieval times. There were so-called quack doctors. They quack like ducks when they're describing their products. And they had various ideas, various concoctions that they sold to people who were desperate for this. They got very few people coming to complain because they weren't as effective as they should be, perhaps, but they made a lot of money. Here's a more recent example of a less rational design. There's this not very distinguished paper in the UK called the Daily Mail Online. I just wanted to point out this article there. Happy pills wards off Alzheimer's. Widely used antidepressants can help reduce formation of the plaque. US academics admit it's too early to recommend the drug to patients, and the experiment was conducted in healthy adults between 18 and 50. And amazingly, none of these people get Alzheimer's disease. They wouldn't anyway, but that was a great, complete success in their trial. So how do we do this more rationally? And we've done a huge amount of work in this that I'm not going to talk about, but I did want to put this equation up, don't faint at seeing it, which is that what we've been trying to do is to understand the detailed mechanism by which the molecules clump together and end up forming these fibrils. And there was a huge breakthrough by a man called Thomas Knowles, who went from a graduate student to a full professor in Cambridge in 7 years, which I think is a record. And what you do is you take the experimental data, these are the points here, this is time, and this is how much aggregate forms. And this is another example, time against how much aggregate forms. And what he realized is you could break down this process into steps. This is the first sort of nucleation, a couple of molecules clumping together. They can then grow as more molecules come in. And they can also fragment, actually. And if you fragment them, you get twice as many aggregates, so they grow twice as quickly. And this is why you get this rapid proliferation. But what was most important is he could write down the steps of these microscopic processes. And then he did something which we all thought was impossible. He developed an analytical approach to get the rate constants of all these processes. So now we knew the mechanism, the detailed molecular mechanism by which, say, the Alzheimer-related peptide converts into these toxic aggregates. And on the basis of this, we've tried to design a rational drug discovery strategy. Involves Sarah Linser from Lund and colleagues in Cambridge. And the idea is, again, I'm missing out about 5-10 years' work, is we come up with candidate molecules, a library may have 50,000 compounds that we think might inhibit these processes and change the rate, make the aggregation slower. And then we look at the mechanism. We look at, do they stop forming these toxic species? Can we look at the mechanism? And can we evaluate their effects on toxicity? And this is just one example of how effective this can be. This again is the A-beta system. This is forming aggregates. You can see it happens quite quickly. There's a lag phase where nucleation occurs, and this rapid growth from proliferation mechanisms. And if you take a small molecule from this library, you can see this can slow this down dramatically. And other molecules have huge potency. So the aggregation here, effectively at a 5-fold excess, aggregation does not occur at all. And so you get this massive effect. So these molecules are able to inhibit dramatically the key steps in the aggregation process. And we've also looked at antibodies, for example, looking very carefully at how they affect either these nucleation processes or the growth processes. We wanted to test these in model organisms. It's not very easy to put them into people. You can't even put them into your graduate students. So we used a model organism, the nematode worm, which has been used very widely. And we developed a very high throughput screening device. We can monitor 1 million worms a week, so we get very high statistics. I just wanted to show you a couple of pictures of this. And so this is one example. These are worms which have got the protein associated with human Alzheimer's disease in there. They're not very happy. Happy worms move around a lot. These are not. We put an inhibitor in there, one of these molecules we've selected, and they're very, very happy. Fantastic news for demented worms. We can stop the process very effectively. And also you can see it stops the deposits. These are shown by fluorescence probes. And this actually is a common anti-cancer drug. It's too potent to use therapeutically, but it's a drug which has already gone through some of the clinical trials. And so this really reveals dramatic effects you can get, at least in these simple systems, from small molecules which are being designed in this highly selective strategy where we can pick them out and we can improve them. I'm not sure if you can see this, but actually it's not just paralysis that happens. We can look at really important functional processes, like the ability of these worms to go towards an attractant, normally food. I'm not sure if you can see this, but actually The worm populations are put here. This is the healthy worms. These are the Alzheimer model system, and these are with the drug. The worms are put down here in a sort of clump, and the attractant is here. I'm not sure if you can see this, but actually you can see the healthy worms very rapidly sort of declump, and then they head over here. The Alzheimer worms are extremely unhappy, and let me go back to that. And sorry, let me go back to that one. They're not really doing anything at all. They've lost the ability completely to find the attractant. But if you can see it, these are ones with the drug, and they're all heading very rapidly for the food, and they move from here, and all of them get over here. So it effectively completely recovers. It's really quite high order ability of this system to head for to be corrected by the drug. And actually, it's a sorter. It's not really a cognitive function, but you can actually maintain it or recover it by these molecules, which we hope are protective drugs. So I hope I've given you some sort of flavor of going from really academic research by accident into potential drugs. I'm personally very optimistic for the future. I think that we will be able to bring this down. Otherwise, you're going to be paying a huge amount of tax to look after people from my generation. By bringing together a lot of techniques and then developing a rational process for therapies rather than the guesswork which has been used even in Alzheimer's disease by drug companies before these mechanisms were known. And we hope we can combat this really devastating plague of neurodegenerative diseases, which otherwise, as I say, will have an effect on the elderly population of something like the Black Death, where half of the population over the age of 80, 85, which is going to be life expectancy, will actually get Alzheimer's disease with all the properties of it, and they will die from it or die with it. So I often say I did all the experiments myself, but there were other people involved. And this is just a selection of students. They're actually Italy, the UK, Sweden, Germany, Singapore, completely international group. But what's been really special for us is actually to maintain long-term collaborations so we can do experiments which we'd never, ever be able to do. And some of these collaborations go back 30 years. And Fabrizio Chiti in Florence, Sam Cohen in Cambridge, Thomas Knowles in Cambridge, Sarah Linster in Lund, and Michele Vendruscolo are the sort of core team. So I hope I've given you some idea of what could happen in the aging Earth in the future. It's not a happy scenario. It's very expensive. It's going to be incredibly divisive socially and terrible for the people who have it. But I think we need you to recognize that working in this area is absolutely critical. I think it's very exciting. There's lots to be discovered, and I am very optimistic that we will end up with drugs. There may be small molecules, antibodies, which are effective, both in as a sort of statin-like molecule, which you take at your age to stop the disease developing, or some drug which will stop the spread of the disease, rather like anti-cancer drugs are designed to do, so that the process does not progress to the full-scale disease. So it's been a great pleasure to be here. I want to thank Laurie and Bengt again, and thank you for your attention.